working is really in trying to understand the dynamics of these agents as they move through porous materials, also in the presence of flows. We look at this also from a different perspective, where we really think about fluid mechanics, like the hydrodynamic interactions of swimmers with rough boundaries or also elastic deformable surfaces. And finally, we're working on the physics of when elongated active agents move together and how their transport properties change, but also their material properties. And so in this talk today, I'll focus on how we can characterize transport of active agents, but I hope Hartmut will show you some of this other work tomorrow. And so let's revisit and look back at this run-and-tumble behavior of E. coli. So as I said, these cells move straight for a while and then randomly change their direction. And the way this works is the following. So the flagella are attached to their body with a motor. And when the motor spins in a counterclockwise way, the flagella can form a bundle, and that allows the cells to self-propel. At the tumbling event, then one or several of these motors start spinning in the opposite direction. That sort of leads to dissolving of this flagella bundle and the tumbling event. And once all these motors are spinning again in a counterclockwise way, the bundle can reform and the cell can start swimming again. So there's been a lot of work on the molecular basis of about the functioning of this bacterial motor, when it spins clockwise or counterclockwise. And the way this has been studied was by looking essentially at cells that are attached to a glass surface, and then one can simultaneously track the rotation of the flagella by attaching a bead, as well as a concentration of some molecular species inside the cells. And so what has been found in this work is that if phosphorylated QY, and that's only one of two components I'm going to mention here today, is present in the cell at a high concentration, this bias of clockwise rotation approaches 1. And so what that essentially means is if this phosphorylated QY is present in the cells are expected to tumble. Okay? And so I'm really not an expert on this chemotactic signaling pathway, but there's a second protein that plays a role here, and this is QC. So now QC essentially this phosphorylates QYP, and in that sense is expected to suppress the tumbling behavior and lead to swimming of the cells. And so looking back at the picture I showed you before, it seems that we need to express QC, or cells need to express QC, to actually transition from a tumbling phase to a running phase. And while this has been studied, let's say, in terms of the motor, there hasn't been made any relation between the molecular mechanism of the motor and the swimming at the population scale. And one of the difficulties is essentially, at least from my point of view, is that it's hard to quantify rapidly these tumbling statistics of bacterial cells, because they move in 3 dimensions and they move relatively fast. So before I go into detail or into the work, I'd like to acknowledge the people that were involved. And this is a project that has been going on for a long time now. So I'd like to mention in particular Vincent Martinez, who is in the group of Wilson Poon at the University of Edinburgh, who did the experiments. On the theory side, we worked together with Yongfeng Zhao, Thomas Franusch, and Julien Tailleur. And the strain that I'm going to talk about was provided by Nansu from Xi'an University. And so, as Roberto pointed out, we're going to be lazy here, so we're using DDM to characterize swimming dynamics of cells in 3 dimensions, because tracking them is very difficult and takes a long time. And so DDM, as Roberto already said, relies on taking images or movies of your samples, looking at the differences of those images and then correlating them in Fourier space. I don't think I have to say much more now. I just want to say that the output of DDM is essentially related to this intermediate scattering function, as we've heard earlier today. And what's really, so the intermediate scattering function is essentially defined in this way. It's the average of the exponential of minus ik, where k is the wave vector, times the displacement variable. Okay? And so one difficulty perhaps also with this method is that if you want to learn something about your experimental measurements, you also need a theory to compare with to actually learn what's going on in your sample. And so, as I said, as Roberto also pointed out, is that this function allows us to measure dynamics at different length scales, resolve dynamics at small as well as large length scales. And mathematically, it's a characteristic function of this random displacement variable, so we can actually look at it analytically. So I'll just show you perhaps some work where DDM has been used to actually study the transport properties of microorganisms. And so in the group of Wilson-Poon, they did a lot of work on looking at swimming E. coli bacteria, but also at sperm cells and amidomonas. And so what they were able to extract by using DDM was the swim speed of those cells, the translational diffusivity, and also the amplitude and frequency of the oscillatory motion of sperms and chlamydomonas, which move sort of in helices when they move in 3 dimensions. And so these are pictures of these graphs of these intermediate scattering functions, and I'm gonna explain to you them in a minute. So we have recently also looked at this for synthetic colloids. So we looked at Janus colloids, and here again you see these intermediate scattering functions now for different wave numbers. So we resolve essentially dynamics over several length scales, okay? And this allowed us really to characterize the dynamics of these Janus colloids in terms of this active Brownian particle model, and we could resolve this transition from active motion to large-scale, like effective diffusion, because these particles perform rotational diffusion. So what hasn't been done to date actually was accessing actually tumbling statistics of swimming microorganisms, and one reason is it's quite hard experimentally to measure dynamics over long times and large length scales, which you need to actually resolve the tumbling behaviors. And also there was no model available to make sense of experimental data. So let me start with introducing you a simple model for the run-and-tumble dynamics. So we essentially think of cells that move straight for a while, and then they tumble randomly and also into a random direction. And so at large scales, we know this leads effectively to a random wall. So the theory that we use is the renewal theory, which you can see here. It's described in terms of these 2 equations, one for the probability that the cells displace a distance r during time tau, sorry, this one, yes, and one for the probability that the last tumbling event happened at an earlier position, or at r time tau. And so the first equation here contains the probability that the cells are at position r at time tau without ever having tumbled before. So now that takes into account that the system that we consider is essentially in a stationary state. And then we have the sum over all previous tumbling events. So here this is essentially the probability that the cells have tumbled at an earlier position at an earlier time, times the probability that the cells keep running at that time, times the propagator that describes the dynamics during the run phase. Okay, and so the second equation also contains sort of the probability for the first tumbling event, and then the sort of the sum over all other tumbling events. And so we have the probability that they have tumbled before, times the probability that the run ends exactly at a certain time, times this propagating. Okay, so one thing you may note when you look at these equations is that you have essentially 2 convolution products, and we can move to Fourier space to make these equations more tractable. And so we have these 2 equations which are decoupled in terms of k, so they can be solved independently for each k. And they're much easier than in real space. And also the Fourier transform of the probability density, as we learned in the morning, is essentially now our intermediate scattering function. So what we can do now is we can solve these 2 equations numerically and compare those to experimental observations to learn about the swimming dynamics of cells. So now reality is a bit more complex in the biological world. So we always have, when we look at these samples, fractions of non-motile cells. So those that are essentially dead. So we have to account for these too. So, and we assume that the non-motile cells are just diffusive. What's also important is that E. coli need a certain amount of time to tumble, and that has to be accounted for in the theory as well. And then finally, cells display a non-negligible velocity distribution, and so we have to also include that in our model. So that leaves us essentially with 4 equations, 2 that describe the run phase and 2 that describe the tumbling phase. And I just want to say, I don't want to go into detail, but for now, here it's just the propagator, so we can decide on how the cells move in either of those phases. So in the run phase, we say they just move straight, and there's a certain velocity distribution associated with it. And in the passive phase, they are diffusive. And another assumption that we make is that the run and tumbling time distributions are exponential, but I should say that the theory is not restricted to that. It's just restricted to run and tumbling time distributions that have a finite mean run and tumbling time. And so then from that we can compute the full expression of the intermediate scattering function and compare that to experimental results. And so before we go to the experiments, I just want to point out a few features that are now different to the simple passive case that we saw in the morning. And so what you can see here are intermediate scattering functions, again for different length scales as a function of time. And so if you see these oscillations, that's a signature that the cells are swimming. So that's a signature for an out of equilibrium process. If you see something in the scattering functions that has like a plateau, that corresponds to the fact that cells are tumbling. And then we also see this nice exponential decay at large length scales, because that means our system becomes effectively diffusive. But we can also see a plateau still at the end, and that corresponds to motile cells, because for those these functions decay slower. Okay, and so now, before we could actually fit all of that to experimental data, we established a fitting protocol, if you like, but I'm not going to go into details. So if you are interested, we can talk about that later. So let's now jump to the experiment. So we looked at dilute samples of E. coli cells moving in bulk. So we were able to resolve length scales from about 1 to about 400 micrometers, which is really important because of we have to resolve several tumbling events. The experiments average essentially over 10 to the 4 to 10 to the 6 cells, but it's still dilute enough so they are not interacting. And so this is what the result, these are our results essentially. The scattering functions are just shifted so we can see the different features. And what you see, first of all, is that we find very nice agreement with our renewal theory. Okay, so the lines are the theory, symbols are experimental results, and we see some hint of these oscillations at small length scales due to active motion, but we see that these disappear as we move to larger length scales because motion becomes randomized due to tumbling. Okay, and so now this fit actually gives us access to motility parameters of E. coli cells, so we can measure the speed, the translational diffusivity, as well as the fraction of passive cells that has been done relatively well before. But now we can also access the run times of the cells and their tumbling time. That's quite new, and that's also really not so trivial. But so now you might ask whether this is consistent with previous work, or also with how can we check that this makes sense. So we can first of all look at the theoretical prediction of an effective diffusivity by plugging in the fitting parameters, and we find the effective diffusivity is about 200 micrometers per second. And we can extract it also by looking at these intermediate scattering functions at even larger length scales and look whether they decay exponentially, and then we can sort of extract the effective diffusivity just from these two curves. And also in that regime, we see that so by doing that, we come up with an effective diffusivity that is very much in agreement to what our model would predict. And just to convince you, so these are experiments from the 70s from Howard Berg's lab, and they found parameters that are like tumbling times that are slightly smaller than what we see, but overall the fraction of time running that they find very much agrees with what we also find. So overall, we think we're quite confident that what we are doing here is actually making sense. So now that we have validated this method, we can come back to this question if we can relate at least some aspects of the bacterial motor to large-scale swimming dynamics. And so to study that, we actually look at the strain of E. coli where we delete the KISI gene. But instead of that, we introduce or non-introduced an inducible plasmid. And what that means is if the cells are grown in an environment where there's IPTG present, they are able to restore the expression of QC. And so the hypothesis is that if cells are grown in an environment with a low IPTG concentration, that they would be tumbling all the time because they don't express enough QC. But if they are grown in an environment with high IPTG, they should resume swimming. And so to study that, we looked at the intermediate scattering functions again. And so here you can see experimental results for different wave numbers. So let's just focus on this plot here. And here you can see that for different IPTG concentrations, right? And we see that if you a change in the curves, right? And we see that as IPTG increases, we actually start seeing these oscillations, and that is a fingerprint of the active motions of the cells, right? And for IPTG 0, we essentially see a relaxing exponential, which is reflective of simple diffusive behavior. And so we can do better than just looking at those curves. We can actually extract the motility parameters from the experimental data, and what we see is the following. So we indeed find that the speed of the cells across different IPTG concentration remains constant, but what does change is the runtime. So we see that we can tune now the runtime as a function of IPTG, and it increases from roughly 0 to about 6 or 7, and then we can also look at the product of those two, and we see an increase of the persistence. Okay, we did that for 2 different biological reactions. Yes. Now we know that the persistence is as a function of IPTG. So now what about the expression of QC, right? So we can independently measure the concentration of messenger RNA of QC per cell, and we do see that as a function, it increases as a function of IPTG. And so now this allows us to actually correlate the persistence length with the concentration of messenger per QC. So we use now DDM essentially to learn something about also the functioning of the bacterial model. And so now a natural question that comes up perhaps next is like, how can we understand drift of the behavior of bacteria when they are in external fields, right? And so what we were interested in was actually looking at how cells behave when they move, for example, in a chemical gradient. So there's sort of a preferred direction of motion. Similarly, when many cells respond to chemicals or to signals that are emitted by other cells, and that can also tune their tumbling behavior. And so there's another change of orientation due to an external field. And then also in the field of synthetic active particles, so you can see here that if the rotational diffusivity of the particles is changed, you can also see interesting dynamical behaviors. And so what we are, so the fact that in active method translation really couples to orientation leads to interesting dynamics, and we wanted to sort of establish a minimal model to study such systems. And for that, we looked at the framework of stochastic resetting, but now instead of resetting the position of the particle, we reset its orientation. And so what that means is, and this is really PhD work of Yanis, so what we look at is essentially active particles that move are similar to an active Brownian particle, and at random events in random time, they start to reset, or they reset their swimming direction. And that's sort of like a minimal ingredient to achieve directional motion, like an overall drift of the particle. And so in this minimal model, we have one important dimensionless parameter that essentially couples the resetting rate, like the frequency at which those agents reset towards a specific target, with respect to their rotational diffusivity. Okay. And so now for this model, we can actually, or we can reformulate this also again in terms of renewal equations, and that I've shown you before, but now we have to take into account a bit more carefully the angular dependence of these equations. And so you see they look fairly similar apart from this term. And this term essentially now includes a probability of the angle after a tumbling event. Okay? So for the run-and-tumble particle, this would be essentially random, so it's a uniform distribution, but now we have a delta distribution. And so what we can actually do, we can solve this in Fourier space analytically in terms of Mathieu functions, so we've done that before, and that then allows us to rapidly extract intermediate scattering functions, for example. And so I just want to show you like a few qualitative features, and then I'm essentially done. So if we have lambda which is smaller than 1, that means that active particles can actually reorient due to rotational diffusion before they become aligned again with the external field. So we see this signature in the intermediate scattering functions again, as we see sort of a decrease of oscillations and then an increase again. And so that sort of reflects that at large scales we have a drift due to this chemotactic behavior, if you like. And here you just see if lambda is larger than 1, that the oscillations just keep growing because particles move along a specific direction. And perhaps one point to take home from this is also that if there is a drift in the system, your intermediate scattering functions become complex, and that's a signature of this type of dynamics. Okay, I think with this I just want to conclude. So I hope I could convince you that using a lazy technique, we can actually extract motility parameters of E. coli cells, and I introduced a strain where we can actually now really tune the tumbling statistics irrespective of the speed of the cell, but not the speed of the cells. And we can now move towards models that start taking into account external fields, and so we can study chemotactic behaviors of very fine, very quickly. Just we are looking for PhD students as well as postdocs at the Max Planck Institute for the Physics of Complex Systems. So please spread the word. And with this, thanks for your attention.